Hi, everyone. Welcome to the Physionic podcast. My name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. And the real credential here that I should be mentioning is that I am a researcher in an autophagy and apoptosis laboratory. We focus on mitochondrial work, but that mitochondrial work is based around autophagy. So that is actually the topic of this particular podcast, which I'm really excited about because this podcast is going to be centered around a paper that my lab just released, and I am one of the authors of that paper. So this is actually my first publication in autophagy specifically. I have other publications in metabolism and muscle research and things of that nature. But this is the first one in autophagy since I've joined this lab. So this paper has been a work in progress for quite a while, quite a long time. Over a year of work has been put into this. And the lead author, Yumiko, and my boss, Marius, certainly took the brunt of the work. And I came in near the end and did some writing as well as some of the research that kind of needed to be done to clean up the study so that it could get approved by the journal that ended up accepting it. So I'm going to go and throw up the name of the study, which I realize is going to be gibberish to most of you. And it's called Parkin Independent Mitophagy via DRP1 mediated outer membrane severing and inner membrane ubiquitination. Okay. Again, as if you're familiar with how Physionic runs, don't be scared by that title. I'm going to be breaking things down as well as I possibly can. Use my teacher hat and put it on and see if I can walk us through some of the research that we ended up going through and to get to actually a really cool conclusion, which kind of in a general sense is a new way of looking at autophagy, specifically in its relation to mitochondria. So what was originally thought was that there was this one primary pathway for autophagy, and that still exists. We're not debunking any of that. But we end up introducing a new pathway of autophagy. So it's a pretty impactful paper in that regard. Of course, if you talk to any researcher, they're all going to say that their paper, their study is highly impactful. But let's go ahead and jump into it. If you're listening to the podcast, don't worry, as always, because I'm going to be describing things. Of course, I'm going to have data that's going to be jumping up on the screen, but I'm going to try to keep things relatively general as I go through so that you don't feel lost as you're going through. But if you would like to see actual visuals of some of these things, because it is going to be a lot molecular biology based, cellular biology based, then feel free to jump onto the video podcast where you're going to see a lot of these graphics and stuff go up. So let's start with a really high level education right now. Let's start with kind of understanding what are mitochondria, why are they important? So mitochondria are within most of our cells. We do have a subset of cells in our body that actually don't have mitochondria, and they produce energy through other systems that aren't mitochondria mediated. But I kind of just alluded to it that our mitochondria do produce energy. That's their kind of, I suppose, main function. And you'll hear this many, many times, considered the powerhouse of the cell. Well, that idea is because it's producing adenosine triphosphate. It's producing cellular energy. That's essentially all you need to know. However, mitochondria actually have other functions as well. They regulate the health of the cell. They are one of the primary mechanisms by which cells end up deciding, in a manner of speaking, if they're going to end up killing themselves. And cells do this for good reason, to avoid cancer, for example. So if too much damage is occurring to the cell, sometimes it can use a system called a back system, which is these two proteins that end up puncturing the mitochondrion, and then you get damage that ends up leaking out into the cell. And if enough of that extra damage occurs, then the cell ends up losing all of its ability to produce energy, and therefore it ends up killing itself, or it ends up dying, I guess I should say. So that's one of the other functions that the mitochondrion has, but that's not really what we're going to be going into in this podcast. So just know that for now that you have cells, and inside those cells you have this organelle known as the mitochondrion, and it doesn't really matter the function too much because we're not so much focused on the function in this podcast, but just know that it has particular functions, and they're pretty important. Cellular health is one of them, the survival of the cell from an energetic standpoint, and it has other functions as well, like creating iron clusters and things like that. But again, no need to worry about that. Okay, so now something that's needs to be taken care of when it comes to mitochondria is their regulation. So you need to make sure that the mitochondria that you have are healthy, right? It's not like they have emotions or anything like that. So I don't want you to think about it from that standpoint, but think of it like a car. You want your car to be functional. You don't want certain pieces falling off. And maybe it still works when you have certain pieces falling off. Maybe the windshield wiper falls off. But if the engine is broken, then it's not going anywhere. And the cell is constantly remodeling itself. It's constantly trying to make sure and maintain and constantly checking if all the mitochondria within it, because you don't just have one, you have many, many mitochondria in a single cell. It has to constantly check that every single one of those mitochondria are functional, that they are producing what they should be producing, that the proteins that make up this organelle are functional, that they are correctly designed, that they're correctly put together, and that it's serving the cell as it should. So, as a result, it has two different systems, the cell has two different systems to regulate the mitochondria and make sure that the health of the mitochondria is where it needs to be. And these are, if you've ever heard of the term catabolism, that is what these are. So they are catabolic systems. They break down functional or just kind of put together proteins to non-functional or just kind of their constituent parts. So they degrade proteins from their put together part to their unput together part, if you want to put it that way. Kind of like, let's go back to the car example. Your car is put together, and if you take it apart for scraps, then it's not put together. And that's what the cell does. So it has two different systems by which it does that. The first system is one that we won't really be discussing, but I'm going to mention it here. It's called the proteasome system. So the proteasome is a protein that looks like a helix, and when proteins interact with it, the proteasome will essentially disassemble those particular single proteins. And those single proteins then get broken up into what are proteins made out of? Maybe you already know the answer to this, amino acids. So it breaks them up into dipeptides, tripeptides, as well as single amino acids. Dipeptide being 2 amino acids and tri being 3 amino acids. So that's one system. So if mitochondria maybe just have a little bit of damage on, a few proteins on its outer surface, then the proteasome will come in. You have different systems that come in and just kind of pluck those particular proteins off of the mitochondrion instead of getting rid of the entire mitochondrion, because that's very wasteful. So you would replace, let's say, the windshield wiper on your car. You wouldn't replace your entire car if the windshield wipers don't work. And if you do, you're a real baller. I would love to have your kind of money. But you're also incredibly wasteful. So that being the case, the proteasome has that function. It's just going to pluck out some particular proteins that need to be destroyed, and then the cell ends up recycling some of those amino acids, and the rest of them can also be disposed of. But the second system, however, is the one that we're interested in, and you've likely heard of, is autophagy. So autophagy in this system is a way that the cell can essentially clean itself up. So if a mitochondrion has a ton of damage to it, a lot of damage, then in that instance, the cell will create what's called a phagosome. And this phagosome is just kind of a lipid structure, so it's a fat structure, a vesicle that surrounds the entire mitochondrion. and then the damaged mitochondrion. And once it closes, then what typically happens is you have the fusion with what's called another vesicle, so another kind of sac-like looking thing, and they fuse together, and you have this phagosome that has the mitochondrion in it, and I mean, it's just trapped this mitochondrion. Okay, great. But what it needs to do is actually integrate a kind of destructive enzyme-filled, another sac filled with kind of destructive things. So it needs to actually have the workers to actually destroy the mitochondrion, and that is called the lysosome. So once both of these vesicles, one containing the mitochondria, one containing different enzymes that destroy proteins, and also a lowered pH, so it's a highly acidic environment. Once those two fuse, then you have the ability to actually undergo full-on autophagy and destroy the mitochondrion. And it's not like the cell is going to just dispose of all of the constituent parts that it gets from the mitochondrion. It's going to recycle some of those to maybe create new mitochondrion, or it might create other proteins from those amino acids, or it's going to end up getting rid of some of them. So those are the two systems. You've got a small-scale proteasome that picks apart single proteins, and then you have this autophagy, this phagosome and lysosome system that ends up engulfing entire organelles. And in this situation, we're concerned with how it affects the mitochondrion. Now, typically, this system of the autophagy system and its relationship to mitochondria is changed in terms of what it's called. It's called mitophagy. So if I use those terms interchangeably, all I'm saying is that mitophagy is autophagy of mitochondria. That's it. They are the same. Autophagy and mitophagy are the same. It's just that all of mitophagy is autophagy and not all of autophagy is mitophagy, if you want to have a logician's argument to this. However, there is what's considered a canonical pathway or a traditional pathway for understanding how autophagy works. So what researchers try to do is they try to figure out what are the molecular mechanisms, what are the proteins and molecules that have to interact to allow autophagy to actually occur. So what attracts autophagy to a mitochondrion, for example? If you have damaged mitochondria, if you're older, if you have Parkinson's, if you have dementia, whatever it might be, whatever disease, if it's mitochondrial related, how does autophagy end up knowing, oh, that mitochondrion is the one that we need to destroy and not this healthy one over here? So one of the mechanisms, and the traditional mechanism that people have focused on, is called the Parkin or the pink-Parkin pathway. You can kind of use those interchangeably, Parkin-pink pathway, either way. So it's based around two proteins. One of them sounds kind of funny. It's called pink, and the other one is Parkin. It's not actually pink, but in my visual that I've created, I just went ahead and made it pink because, well, it's just easier to remember. So if you're listening to the podcast again, if you want to, you can hop on over and check it out. But it's just a pink protein. That's all it is. So the mitochondrion, to give a little bit more nuance to understanding how the morphology, the look of the mitochondrion, how is it structured? It has two membranes, and I'm going to go over this again in just a little bit just to hammer this home. But it has two membranes. It has an outer membrane, which actually separates the inside of the cell, because again, all these mitochondria are inside of the cell. It has an outer membrane that separates the inside of the cell from the inside of the mitochondrion. So just make that distinction in your head. And the cell, just kind of like the cell, has a cell membrane that separates the inside of the cell from the outside of the cell, which might be filled with what's called extracellular fluid, or even a little further beyond that, the blood system. But we're looking at things even more microscopically. We're talking about within a single cell. So in this situation, the mitochondrion has its own membrane known as the outer mitochondrial membrane. And I've mentioned before that that's not exactly very creative, but it does serve its purpose. You know that it's on the very outer edge of the mitochondrion. Now, there is another membrane, however, because the mitochondria also has a second membrane known as the inner mitochondrial membrane. And I'll touch on that a little bit more in just a second. But let's focus on just this outer mitochondrial membrane. So on the outer mitochondrial membrane, you have a series of different proteins. And I'm just, if you're watching the visual, if you're seeing the visual, it's really basic. I'm doing no justice to the amount of proteins that litter the entire mitochondrial membrane, but I'm focusing on just a few. One of them is called TOM. So you have like TOM 20, TOM 40, TOM 44. I mean, a lot of different types of TOM proteins, and these are outer mitochondrial proteins. That's all you really need to know. They have a particular function. I'm not going to go into them because we don't need to know that. But just know that TOM proteins, if that's the TOM 20 protein or the TOM 40 protein, they're all embedded into this outer mitochondrial membrane. And that's going to be important because as I go through the actual data, that's one of the distinguishing features of how we can tell that we're looking at the outer mitochondrial membrane. So anyway, this TOM protein can be what's called tagged or ubiquitinated, and that's going to be important as well. So what typically happens in the Parkin-Pink pathway is that pink is a protein that will bind to the outer mitochondrial membrane, and then it will undergo, if there's some sort of signal, and we're going to go over that signal because that's what our paper actually goes into. If there's some sort of signal, then pink will undergo a transformation. It will phosphorylate itself, which is called an autophosphorylation event, meaning that we take a phosphate molecule. You don't really need to know what a phosphate molecule is. Just know that there's a change, that this molecule, known as a phosphate, is attached to pink. So pink will attach a phosphate to itself, like putting a top hat on. And in doing so, it sort of starts to draw attention to itself. I feel like I'm describing some people here. None of these are emotionally driven decisions. They are biochemical processes, but I'm trying to make them relate. So then pink, once it has its top hat on, what ends up happening is that Parkin, so again, these two proteins, pink and Parkin will start to ubiquitinate other proteins that are littering the outer mitochondrial membrane. So in this situation, we're talking about TOM. So this TOM protein will start having a bunch of tags added to it, and those tags are called ubiquitin. So ubiquitin proteins are added to the TOM protein. That TOM protein is embedded in the outer mitochondrial membrane. Now, pink, recognizing this chain of ubiquitin proteins, starts slapping phosphates onto those ubiquitin proteins. And that entire series leads to a recognition of those proteins, of that kind of conglomeration of pink, phosphorylation, the ubiquitin added by Parkin, and the phosphorylation of the ubiquitin. Now, if it's just a single protein, maybe the proteasome can deal with it. Remember that's, cylindrical thing that I was talking about earlier that can, deal with individual proteins. However, if you have mass ubiquitination across the entire mitochondrial membrane, across the entire outer mitochondrial membrane, then who do you think comes in? Not Ghostbusters, it's autophagy. So autophagy then becomes a factor. So let me throw this graphic up. So if you're looking at the visuals, I've removed pink, I've removed Parkin, and I've removed the phosphorylation just because I want us to, because I don't want the slides to be too busy. But at this point now, you have Tom that's been ubiquitinated by a chain of ubiquitin proteins, and that leads to the recruitment of another protein called P62. And then it can actually recruit other proteins as well. But ultimately, in a general sense, they're called adapter proteins. So these adapter proteins will bind to either Tom or this ubiquitin chain. And once that binds, that is the signal for autophagy. Once that binds, autophagy, which is highly related to another protein called LC3 will recognize this adapter protein, if that's P62 or if that's something else. So once LC3 binds to P62, you start having the formation of the phagosome. So if this happens across the entire mitochondrial membrane or a large part of it, then this massive phagosome starts to engulf the entire mitochondrion, and that's the process of autophagy. So hopefully I did some justice to it. That is the kind of classical understanding of autophagy. And there's much more, of course, that's involved in all this, but those are the key players that play a role. And again, Pink and Parkin are kind of this classical understanding as well. Now, before I go into the actual data itself, I've probably mentioned this before, but in my master's program, I had this professor that would always ask and always be like, yeah, but where is this happening? And that's why I always point out the mitochondrion is in the cell, and we're looking at a single cell, and we're looking at, really tiny subcellular aspects, because I really want us to understand how how minute some of this stuff is. So just to orient ourselves just one more time, please bear with me here. I'm going to give us some context on the mitochondrion itself. So again, I mentioned that the mitochondrion has this outer membrane and it has this inner membrane. And those actually create spaces between them. So the outer membrane obviously protects from the rest of the cell. But then there is a space between the outer mitochondrial membrane and the inner mitochondrial membrane. And that space is called the intermembrane space. Now, the reason why I'm pointing this out is because as we're going to be going through this research, there's going to be consistency in what we're going to be visualizing and what I'm going to be describing. So remember Tom. Tom is on the outer mitochondrial membrane, and you will always see it on the outer mitochondrial membrane. So the reason why I'm pointing this out is because when we as researchers end up using Tom as a marker, we're not just interested in Tom, we're actually also interested in the fact that Tom is always associated with the outer mitochondrial membrane. So if we image for Tom, if we look for Tom and we find Tom, then we know that we're looking at the outer mitochondrial membrane. That's important. So just keep that in mind. Now, a protein that we are also interested in, and that is found in the intermembrane space, so between where Tom would be located on the outer mitochondrial membrane and where the inner mitochondrial membrane is another protein called cytochrome C. And this serves a vital function for a variety of different things that I will not be going into because they would just muddy this podcast up. So just know that cytochrome C is roughly related to the intermembrane space. However, then we look at the separation, or what's behind the inner membrane. So there's these structures that are found inside the mitochondrion called cristae. And these cristae are a way for the mitochondrion to be a lot more efficient. Again, not really something that I want to get super into, but ultimately they allow for the mitochondrion, because of this rigid, this kind of structure, this kind of folding structure. That's a great way to put it. As a folding structure, because it gives more surface area within a small mitochondrion, that allows the mitochondrion to produce more energy and to have a greater function because it has more surface area to actually be able to use. So that's why you have these folds within the mitochondria. So if you were to ever actually look up a mitochondrion, you would see these cristae. So those cristae were located in what's called the matrix of the mitochondrion, which is the very center of the mitochondrion. So the reason why I'm mentioning this is because while we have TOM that represents the outer mitochondrial membrane, and we have cytochrome C that represents sort of the intermembrane space, we have other proteins that represent the matrix because they are specific to the matrix. So these proteins in this instance is going to be called CLPP. Okay? You don't need to know the function of CLPP, but you do need to know that it is located in the matrix. And we're going to be looking at, I think, one other protein that's also found in the matrix. So the point is that the localization of these things, not the function, not what they do, but where they are is important because the research is going to be using these targets as ways to actually determine and look at the mitochondria itself, because we don't just use light, we don't just use a regular microscope, we have to fluoresce a lot of these proteins so that we can actually visualize the mitochondria themselves. Okay, massive introduction, I get that. This is also a big paper. It's quite involved, and unbiasedly speaking, it's the greatest paper ever. No. It's not, but it is. We did put a lot of great work into this. I'm excited for the stuff that I can add in the future as we continue this line of research. Okay, so that is a background, an introduction. So in this study, we're aiming to see if there's an alternative way by which mitochondria can undergo autophagy. Because I mentioned the pink Parkin pathway. That's already been known. A lot of people research that, and we even touch on it here. Okay, so now touching on the first piece of data. Remember the proteins that I mentioned earlier. If you're listening to the podcast, you may, if you're interested in actually looking at the data for itself, I've got things kind of outlined out for people, and I'm only going to be covering, I don't know, maybe like 10% of the full paper because it's just so much and it can be really overwhelming. So in this first piece of data, we look at microscope images of mitochondria. So we have kind of a big picture of multiple cells that you're looking at if you're actually looking at the images. So we take images of multiple cells. So those black areas are actually nuclei. They are where we contain the genes that are found, or kind of every one of our cells has nuclei, and those nuclei have the genes that hold the information for the production of all the proteins in our body. Now, interestingly, mitochondria also have genes, but they tend to make up less than 1% of our genetic code. The nucleus makes up over 99%. So that's not really the point. The point is that with our microscope, we were able to take images of multiple cells. So you're seeing 4 cells there and the only reason why we can identify that is because we're seeing 4 black circles there because those are the nuclei. But what we're really interested in, and what we actually end up using these probes, these fluorescent probes that shine different colors, is for the mitochondrion. So I mentioned cytochrome C, I mentioned CLPP, I mentioned TOM20. So TOM and TOM20 are the same. And here we're probing for these particular proteins because we want to actually be able to see what happens to the mitochondrion when we induce a particular problem. And this problem is induced by a drug. This drug is called actinonin. So what we did is we added this drug to these cells, And then we observed, how did the mitochondria change to this drug? Now, you might be asking yourself, well, what does the drug do? Well, the drug actually affects the mitochondria's ability to translate, or more appropriately, or more commonly said, to synthesize new proteins. So while most of our genetic material and most of our synthesis of proteins originates from the nucleus, remember, a small percentage is from the mitochondria. So we do have DNA in our mitochondria. And that is the template to produce more proteins. So this drug, what it does is it actually screws up that system, but specifically only in the mitochondria, not in the nucleus. So what it does is it screws it up in a particular way. So it starts producing, the mitochondrion starts producing non-functional proteins. And if we go back to the very beginning of this whole rigmarole that I gave you, with the idea of damaged proteins, damaged mitochondria, what is that going to do? It's going to start to induce autophagy. So in this situation, we add a pretty large amount of this particular drug, actininin, and then we want to see what happens to the mitochondria. So we look at cytochrome C to see the effects on the intermembrane space. We see the effects on the matrix through CLPP, which is the one I mentioned earlier, and finally the outer membrane, which is TOM20. And then if we merge all of those images together, we end up seeing it's kind of a cool, I mean, they're kind of small on screen, but they're this grouping of all these different colors. So green, red, and blue. And all of that merged obviously creates overlap. So then from there, we can actually see what differences there are. And we compare that to cells that we haven't treated. So we haven't added actinin to these other cells. And we just want to see what differences there are. And what we ended up finding was something really interesting. And this started to kind of pique our interest because what we saw is a redistribution of cytochrome C. So that protein in the intermembrane space was moved to different areas within the mitochondrion. So we're looking at multiple mitochondrion, and for some of them, they have really highly elevated levels of this cytochrome C. And if you're looking, you can see that the yellow arrows are pointing those out. Those are the specific mitochondria that have really high levels, I mean, abnormally high levels of cytochrome C. So that's really intriguing. And that's really all I want you to take away from this, that this is weird. Like that's the only reaction I want you to have, that it's really strange. Like some mitochondria have really high cytochrome C, but on the other hand, other mitochondria have no cytochrome C. So something is going on here where this messing up of mitochondrial ability to create proteins, and keep in mind, I'm saying it's messing it up. It's not stopping it. It's just messing up the fidelity, the ability to produce those fully functioning proteins. That is somehow leading to this pretty dramatic change in cytochrome C levels within the cells. Okay, so that was the main point of that first piece of data. Now, the next piece of data is the exact same experiment. We add actinin to some cells and we add nothing to the other cells. And in this situation, however, we're using slightly different cells. We're using what are called DRP1 knockout cells. What does that mean? That means that the mitochondrion, how they typically behave is that they go through what are called mitochondrial dynamics. So they're constantly changing their shape, the way that they function. So one of the ways that it does that is through fusion. So if you have two mitochondria, they can actually tether onto one another. And I wish I had created a visual for this, but they can tether onto one another. and they can fuse together. So the outer mitochondrial membranes will fuse together, and the inner mitochondrial membranes will fuse together to create a single larger mitochondrion. But that's not what we're focused on here. We're focused on this other mechanism, which is the exact opposite. When you have a single mitochondrion and it needs to separate out, so it needs to create two mitochondria. And in that situation, it undergoes mitochondrial fission. So not to be confused with like nuclear fission when you're talking about physicists, but the idea is that you're getting this separation. So with that separation, you have a protein called DRP1. DRP1 will essentially swarm onto the mitochondrion and start to squeeze the mitochondrion, pinch it right down the middle. Well, it doesn't necessarily have to be down the middle, but often it is. So it starts to pinch the mitochondrion to a point where it splits into two mitochondria. However, with the cells that we're using, we've knocked out that protein. So they cannot separate from one mitochondrion into two mitochondrion. And what we find is that they elongate. They're just this single mitochondria is just becoming extremely long. They become very stringy because they can't separate. They want to, but they can't because we've gotten rid of that protein. And what we find is that if you look for elevated levels of cytochrome C, they don't exist. So somehow fission or DRP1, that DRP1 protein, is integral for that effect of actininin on cytochrome C. That's really the big point here. So the takeaway from here, just from these two pieces of data, is that mitochondria accumulate cytochrome C when protein toxicity occurs. And that's what I'm going to call actininin from now. Protein toxicity, the buildup of non-functional proteins. If you think of like dementia, if you think of Parkinson's, I mean, all of these are related to a lot of protein toxicity. I'm not saying that we figured out the sole reason for these diseases, but those have been some lines of thought that have been related to this. So the idea here that you've got mitochondria that are spitting out non-functional proteins. They're trying to create the proteins they need to function, but they're producing non-functional ones because there's this toxicity that's being induced by actininin. So mitochondria accumulate cytochrome C when protein toxicity occurs, and this is dependent on mitochondrial fission because we saw that none of that was happening when mitochondria couldn't undergo fission. Okay, so now this is a really simple piece of data, but it is necessary, or it's at least important. Here, we're looking at a series of different drugs that we ended up using. So actininin is the one that we were using, and we just wanted to know, okay, well, what happens if we just add general stress to the mitochondria? Not protein toxicity, but what if we maybe, stop the mitochondrion from being able to produce energy, to produce cellular energy? Do we see this mosaicism in cytochrome C? Meaning, just to be clear, that you get this elevation in cytochrome C in certain mitochondria and this dip or this complete loss of mitochondria or of cytochrome C, in other mitochondria. And what we found is that with a variety of different drugs, FCCP, oligomycin, antimycin, rotenone, all of those had no effect. It did absolutely nothing in terms of the measure of this cytochrome C strange effect, only actininin. But then when we added actininin plus another drug called chloramphenicol, that drug, what it does is it stops mitochondria from producing proteins. So remember, actininin still allows those proteins to be made. They're just really crappy. They're just non-functional. But chloramphenicol actually stops the entire production of protein. So, and what we found is that it massively reduced the effect of actininin. So now we're getting really good evidence that is specifically the protein toxicity inside the mitochondrion that is causing this sudden shift in cytochrome C. So that's all I want you to know so far. So actininin, protein toxicity, leading to this this effect on cytochrome C. We don't know what the effect is. We don't really know what it means, how it might have something to do with autophagy, nothing like that. We just know that. So only protein toxicity, the takeaway is that only protein toxicity leads to mitochondrial mosaicism. What I mean by that is, again, this change in cytochrome C. So then I skip over a decent amount of data, but of course the paper's linked if you want to look it up for yourself. And then we go from that to looking at some more images, but ultimately we end up changing some of the conditions. So here what we're doing is we're looking at some new proteins. So we're still looking at TOM20 as our measure for the outside of the mitochondria. So the outer mitochondrial membrane. Then we have a new protein for the inner mitochondria, or the inner mitochondrial membrane. And then we have ubiquitination. Now, just to remind you, what is ubiquitination? All I want you to think every time I say ubiquitination or ubiquitin is it's a tag. That's all it is. It's a tag for destruction. So if something is tagged with ubiquitin, it is tagged for destruction, either through the proteasome or through autophagy. But if there's mass ubiquitination, there's mass tagging of, let's say, a mitochondrion, then it's going to go to autophagy. So here we did the exact same thing. We added actininin, and we wanted to know, how does ubiquitination change? Do we see sudden hot spots of ubiquitination? And oddly enough, I've got it marked here, this one panel where you kind of see everything together, because they've got we had ubiquitin in green, we had the inner mitochondria in red, and the outer mitochondria in blue. So obviously those come together to make purple. Then we still see these hot spots of green. So those mitochondria are being tagged by ubiquitin when we add actininin. Now, when we add chloramphenicol, we see that effect is demolished. So we don't see it anymore. And if we add both of them at the same time, we still don't see an effect. So again, this just furthers the proof that this protein toxicity leads to greater ubiquitination of the mitochondria. We still don't know the relationship between that and this elevation or this loss of cytochrome C. But we will be getting to that. So now we're starting to, in this next piece of data, we're actually starting to try to create this link between cytochrome C and ubiquitination. So in this situation, we're looking at the same exact proteins, but here we're using our microscope and we're zooming in as much as we possibly can. And there are more powerful microscopes that can actually zoom in even more, but this is one of the most powerful microscopes. It's called super resolution imaging. And with the super resolution imaging, what we did is we looked at a single mitochondrion. And this single mitochondrion is very round, which is odd. It's different from what it normally would be. Normally, it'd be a little bit more elongated. So with this rounding of the mitochondrion, we want to kind of see what's happening to, let's say, the outer mitochondrial membrane. We want to know what happens to, is ubiquitin all over the mitochondria, or is it just on certain parts of the mitochondria, or are we missing the mark completely? So with these different markers, what we ended up seeing was that ubiquitination was all the way around. So if you just look at the images for ubiquitin, UBFK2, you see that it's all the way around. It's a circle, right? So it's all the way across the entire mitochondrion. Now, if we focus on the outer mitochondria, that marker for TOM20, what do you notice if you're looking at the visuals, is that it's only a semicircle. So something happened there. That means that TOM20, that the outer mitochondrial membrane most likely broke open. So now only half of the mitochondrion is covered by this outer mitochondrial membrane. That means that the inner mitochondrial membrane is now open to the rest of the cell. Because again, the outer mitochondrial membrane is the membrane that protects you or protects the mitochondria from the rest of the cell. So, and we see that ubiquitination, that tagging for destruction, is not only on the outer mitochondrial membrane, but also on the inside of the mitochondria. And this is actually a novel discovery. This is something that our lab has been really focused on. So this part alone is already pretty interesting, but then we ended up trying to relate that to, well, cytochrome C. So we did the exact same type of experiment, but instead of looking at ubiquitination, we looked at cytochrome C. And what we found is that in any mitochondrion that lost their cytochrome C, that were completely dark, could not see any cytochrome C, but we know the mitochondria are still there because we're imaging for the inner mitochondrial membrane as well as the outer mitochondrial membrane, and they're both still there, but then when we switch over to cytochrome C, it doesn't exist. In that situation, we still see this breaking open of the outer mitochondrial membrane. And this is most likely, and this is just kind of speculation, we're not entirely certain on this because we would have to run a little bit more specific experiments on this. The idea being that cytochrome C gets released, gets dumped out. It just floats out of the mitochondria because TOM20, or because the outer mitochondrial membrane, is breaking open. So obviously everything that's in that intermembrane space, that's why I was harping on this point that we need to know where these three proteins are: TOM20 on the outer mitochondrial membrane, cytochrome C more so in the intermembrane space, and then some of the proteins that are on the inner membrane and the matrix. So cytochrome C is leaving the mitochondria, which actually has some pretty interesting implications in cell death, but I'm not going to be touching on that in this podcast. Okay, so that was a lot of information, but the takeaway here, if we kind of pull everything together, is the outer mitochondrial membrane is severed. So that is one of the first times that we've ever seen that. And there's still ubiquitination or tagging of the mitochondria all around. So the outer membrane as well as the inner membrane, implying that the outer and inner membrane of the mitochondria are ubiquitinated or tagged. Now, I decided not to throw any images up here for this next piece of data, but essentially we used an experimental method called a mass spectrometry. It's always hard for me to say. Mass spec is the shortened version. And ultimately what it does is it allows us to identify particular proteins that are changed in an environment, in a particular context that we're interested in. So obviously you can imagine we took actinin treated cells and we took non-actinin treated cells, and we ended up trying to see what kind of differences there were. And we were more interested in, are any inner mitochondrial membrane proteins ubiquitinated? Because normally they wouldn't be accessed, right? They couldn't be accessed if the outer membrane is protecting the proteins on the inner membrane from being accessed by, could be Parkin, could be any other protein that adds ubiquitin, because Parkin isn't the only one. There are other what are called E3 ligases. Those are the proteins that add ubiquitin to these different proteins. So if the outer membrane were intact, then there would be no way that you could have ubiquitination of the inside of the mitochondria, the inner mitochondrial membrane. However, what we ended up finding is that there were proteins that were ubiquitinated, specifically in the condition with actinin. So just confirming everything that we've been talking about so far. So again, I'm not going to show any of that data. We also did what are called pull-down assays to end up to actually prove that the information that we got from mass spec as well. So the big takeaway here is that inner mitochondrial membrane proteins are ubiquitinated, and that's important. Now, we are kind of rounding out near the end here, as we're going to be tying this into autophagy now, or starting to. So now what we're doing is we're looking at particular proteins. So we're looking at this mitochondrial ubiquitination, just like we did in all the other experiments. However, and we're looking at a single mitochondrion. So if you're watching the podcast, you can see actual images. That is an actual image of a mitochondrion. It might look kind of blurry and whatnot, but I mean, we're talking subcellular components here. So our microscope work is getting better and better by the decade. This is actually pretty impressive technology. So in this situation, we're trying to relate ubiquitination and this breaking, the severing of the outer mitochondrial membrane to the protein LC3. Now, if you'll allow me to go back and remind you what LC3 is. LC3 is found right here on, it makes the connection between ubiquitinated proteins on, let's say, the mitochondria, and then the adapter protein binds to those ubiquitin molecules, and then you have LC3 there. LC3 is a great marker of autophagy. So that's what we ended up looking at, going back to the data. So we end up staining or end up looking at this fluorescent LC3 protein, which is fluorescent green, and ubiquitin is blue, and TOM20 for our outer mitochondrial membrane is in red. And here we find again that you have this severing of the outer mitochondrial membrane. You still have the ubiquitination of all the entire mitochondria, including the inner membrane, and you see LC3 is also found in that location. That implies that autophagy is occurring. However, then we also ended up trying to use an autophagy inhibitor, so because that would allow us to kind of stop everything in its tracks. So LC3 might be able to find its way to the mitochondrion, but it's not actually going to be able to go through with its autophagy function. But yet we still see that LC3 is associated with the mitochondria. So, and we wanted to take things a step further. So we end up looking at these cells again, and we wanted to look at these mitochondria and see how does that relate? How do these autophagy markers like LC3, like P62, which is that adapter protein, which is also called the sequestosome, if you want to get real fancy with it, how do these relate to this change in cytochrome C that we talked about at the very beginning of this data? So this loss of cytochrome C is associated with the severing of the outer mitochondrial membrane. We saw that earlier. It's confirmed here again. But where there's this severing of the outer mitochondrial membrane, we find that P62 goes to that area. So an autophagy-related protein goes to that section. So we're now getting this relationship of low cytochrome C mitochondria are now related to autophagy machinery. So that is a signal right there that autophagy is recognizing this change in cytochrome C, and that is leading to autophagy, or the removal, the destruction of these particular mitochondria. Okay, so I don't think I have the data for the next figure, but that's all right. So next, we also looked at cytochrome C levels, and I already mentioned that. Never mind, I already mentioned that. Talking about cytochrome C and its relationship to autophagy. Okay, so the takeaway here is that opening of the inner mitochondrial membrane is associated with autophagy markers that are present, which induce autophagy of that particular mitochondria. And now the final pieces of data are to reintroduce, and I probably should have mentioned this earlier, that none of the cells that we looked at have parkin in them. So they don't express parkin. So we know that all of this stuff, everything I've described so far is parkin independent. And that is the novelty of this particular study, because we're looking at something that's non-classical. We're looking at cells that don't have parkin. Therefore, none of this can happen through that classical pathway. So we end up reintroducing parkin in this particular situation. So we end up quantifying the amount of high cytochrome C in the cells with and without parkin that's transfected. So what does that mean? That means that we end up infecting the cells with parkin. What does that mean? Which sounds kind of weird. We take a virus, or we're not talking COVID. We take an inert virus, or we take what's called a plasmid, which is not a virus, and that will actually infect into the cells. And what that virus and or plasmid contains is the genetic material for parkin. So once we infect the cells with this, and this is essentially genetic engineering of these particular cells, those cells will start to express parkin. And we can verify this by adding a tag to parkin to make sure, okay, yes, we're certain that parkin exists now. We can do it before the transfection, and we're like, okay, we don't see anything. And then we do it after the transfection, and we test again, and we're like, yep, there it is. There's parkin. So what did that ultimately lead to? In these cells that we've done all these experiments in that don't express parkin, we're now expressing parkin. What happened? So we quantified the levels of this elevated cytochrome C, and because that's also a part of it with this combination with the low cytochrome C in these cells with and without parkin, and in the normal and autophagy knockout cells. So we took cells that we actually knocked out autophagy. So we got rid of one of the key proteins, kind of plucked it out almost. And now the cells can't undergo autophagy. And we find is that parkin actually reduces the levels of these high cytochrome C mitochondria. So that means that this classical pathway actually gets in the way of this non-classical pathway, this pathway that we're describing right now. However, when we inhibit autophagy as a whole, that stops everything. So we still see this accumulation that actually gets rid of the parkin effect, and it actually still allows us to see these elevations in cytochrome C. It's like the cell is like, okay, this isn't working, parkin isn't working, so we need to go through this other pathway. But unfortunately, both of them end up going into autophagy, and both of them have autophagy knocked out. So it ultimately leads to nothing. But the point is that we just wanted to see what effect that would have. But then the final piece of data that I have is to, and this is probably the busiest, of course, is to see what effect this has on the classical pathway. So we looked at ubiquitin levels, then we looked at phosphorylated ubiquitin levels. Remember that pink phosphorylates ubiquitin and phosphorylates a lot of other key proteins when we're looking at that classical pathway. So what you would expect is that if we're undergoing autophagy through this particular pathway, this classical pathway, that you would see more of this phosphorylated ubiquitin on the mitochondrion. And then we have a marker for the outer mitochondria, which is still that TOM20 that we've been using this entire time. So in parkin absent cells, we see no breaking, or we do see a breaking in the outer mitochondria, and we don't see much localization. It's kind of dispersed phosphorylation of ubiquitin. So we still see the ubiquitination, but we don't see the classical understanding of ubiquitination with the phosphorylation event through pink. So that's kind of dispersed, and we still see the breaking of the outer mitochondrial membrane. The images aren't quite as clear here as they were in some of the previous stuff, so it's definitely pretty fuzzy in this example. However, once we reintroduce parkin, suddenly we still see ubiquitination. However, now we also see phosphorylation of that ubiquitination, because pink is also active in this situation, and we see that the outer mitochondrial membrane is intact. So this shows a distinct difference when parkin is present and when parkin is not present. So it's two different mechanisms to induce autophagy. One of them is, in a manner of speaking, more violent than the other one. It's kind of like a backup system in case one of them ends up failing. So the takeaway here is that parkin presence reduces this new mechanism of autophagy because it preserves the integrity of the outer mitochondrial membrane and just leads to autophagy of the entire system just based off of ubiquitination of just the outer mitochondrial membrane. However, when parkin isn't present, the cell maybe tries to find parkin or tries to use parkin, and it can't. So somehow you get this buildup of cytochrome C, and then the outer mitochondrial membrane bursts, or it blasts open, and you get the loss of cytochrome C, or that's some of this is based on speculation. We're hoping to do the experiments for this. You get the loss of cytochrome C, and then with that, you also introduce the inner membrane to the rest of the cell, so you have other proteins that come in and start ubiquitinating the inside of the mitochondria. So this is so much information. If you're with me up to this point, I completely recognize this is wow. I mean, it's really heavy molecular biology, but hopefully you're fascinated by how our cells function and how they're able to do this kind of stuff. It's really absolutely incredible. That's why I got into this, or one of the reasons. Okay, so the conclusion, the takeaway from all that, and keep in mind that was like 10% of the data that we had in our paper. We had a lot more. There is a non-traditional pathway by which cells can undergo autophagy that does not involve parkin. I just went over that, but does involve an accumulation of a signal through particular proteins. We're talking about cytochrome C, and I did mention some of the other proteins, but there were other proteins that also accumulated. And the idea is that they accumulate, they create a signal. They kind of create like okay, this particular mitochondrion is about to have some sort of issue, and that leads to this breaking open of the outer mitochondrial membrane. Now, how does that actually happen, though? And I didn't actually go into the data for this very much, but this actually goes back to our fission, to the ability for mitochondria to separate from one mitochondria into two mitochondria. So what we think is happening, what we mentioned in the discussion of this paper, is that the mitochondrion, let's say it has a lot of damage to it, proteotoxicity, these proteins that it's produced, we add actinin in, and then they have a bunch of damage to them. They're non-functional, et cetera, et cetera. It builds up too many of those proteins. It recognizes that. It starts to separate out the functional proteins from the non-functional proteins, in a manner of speaking. And because it can't just be like, okay, well, you just go sit in another room, or in the same room. It can't just have like, okay, good proteins stay in one corner and bad proteins stay in another corner. It actually has to create separation there. So the one way that it can create separation is through fission. It literally splits a mitochondrion with all the bad proteins, just shovels them into a mitochondrion and just splits it, and it shoves that one off, and then it keeps the good mitochondrion functioning. So it's kind of, I don't want to call it intelligently, because these are all biochemical processes, but the healthy mitochondrion continues to survive, and the bad mitochondrion, which is bad off, it sucks for it, but it has accumulated all of these signals of, yes, come kill me, come get rid of me, come eat me, autophagy. And then that is all mediated through fission, through that DRP protein that ends up cutting that non-functional mitochondrion off. Okay, so I didn't really go into that very much, but that is essentially one of the thoughts that we have. So, and that is through the breaking, and eventually that leads to the breaking of the outer mitochondrial membrane. This breaking of the outer mitochondrial membrane leads to the tagging of the inner mitochondrial membrane, as well as the remaining part of the outer mitochondrial membrane. This then recruits autophagy machinery. Now, one alternative thing is the final thing that I'm going to say, which I think is also really, really cool. It's also possible that the cell actually doesn't even recognize the proteins on the inside of the inner mitochondrial membrane. It has never seen that before, because normally mitochondria come in the nice functional two membranes. So the cell never actually experiences the inner mitochondrial membrane because it's constantly separated by the outer mitochondrial membrane. So with the outer mitochondrial membrane, it's possible that it's recognizing that and it's fine with it. Like, we've survived millennia, we've survived, however long with that understanding between the cell and the mitochondrion. However, once that outer mitochondrial membrane ruptures, the inner mitochondrial membrane is not recognized by the cell. So it's possible that it's not ubiquitinating particular proteins because it's like, oh, I recognize that and I recognize that and we need to destroy this and all that. It's possible it actually suddenly sees the mitochondrion as an enemy, as something other than itself, which is called xenophagy. Xenophagy, I don't know, however you want to pronounce it. And that is how we actually fight viruses, and that's actually how we fight bacteria. When they infiltrate into our cells, the autophagy machinery gets recruited to those things because the cell recognizes that is not, I did not create that. And it will then engulf that and destroy it through the lysosome that I mentioned earlier, just bombarding it with stressors, creating, dropping the pH, making the environment a lot more acidic, attacking it with all these proteases and whatnot that end up like just picking apart different proteins and destroying them. So it's possible that the mitochondria is being recognized, and it's also possible, something that we haven't tested, is that the mitochondria is suddenly this foreign object once the inner mitochondrial membrane is exposed. Okay, folks, that's it. That's what I've got for you. Wow. I mean, that was a ton of information, but it's only once every few years that a researcher can actually publish a paper, especially when it comes to molecular biology. So it was great to actually get a paper out there so I can finally, I mean, I've been working in an autophagy laboratory for a few years now, but it's great to finally say I am published in the Autophagies. So that's what I've got for you. If you've gotten to this point, I mean, kudos to you. Hopefully I did some justice to try and explain this. I mean, we're talking molecular biology. It gets really, really complex. I leave out 95% of the information that I could be going over, but I do go into a decent amount of depth. So don't feel like the information that I'm giving you is so incomplete that if you were to go have a conversation with a researcher or someone else, that they wouldn't be like, wow, this is absolutely correct. You know a lot more than the general population, if you were able to glean a lot of information from this. So hopefully I did some justice to it. With that, I wish you a wonderful day, and I'll hope to catch you. Hopefully I haven't chased you off. I'll hope to catch you in the next one. See ya.